Good morning children. Today we are going to discuss about biotechnology. Biotechnology using scientific methods with organism to produce new products or new forms of organism. Any technique that uses living organisms or substance from those organisms to make or modify a product to improve plants or animals or to develop microorganisms from specific uses. See, the biotechnology, the engineering, genetic engineering, in vitro cell culture, protein engineering, bioprocess technology, using for healthcare, diagnosis, immunology, energy and environmental management, food processing, chemical energy, biochemistry, genetics, cell and molecular biology, fermentation technology, agriculture crop improvements, microbiology.
Objectives of biotechnology. Production of such products whose availability in is inadequate or not available. Development of new techniques to minimize the cost of industrial process. Production of safe and high quality products. The main object of biotechnology is to exploit the resource judiciously for the welfare of human society. The traditional use of plants and animals is not included in biotechnology, like agriculture, horticulture and animal husbandry are already in existence for such aspects. History of biotechnology. The term biotechnology was coined by Hungarian engineer Kari Ericki in 1901. It became well established as a branch of science at the end of 20th century. The structure of DNA molecule was given by Watson and Crick 1953. Warner Arbor 1971 discovered a specific enzyme called restriction enzyme from bacteria. This enzyme can cut the DNA thread at appropriate point. In 1973, H. Chohan and H. Boyer extracted a specific gene from a bacteria and inserted it into another bacterium with the help of restriction enzyme. It helped the beginning of recombinant DNA technology or rDNA technology. The technology is the basis of genetic engineering. With the help of recombinant DNA technology and desired gene from any organism can be removed and further it can be transferred or inserted into another organism. The gene transferred by this technique is called transgene, and the organism in which transgene is transferred is called a transgenic organism. In 1976, Baltimore transferred the growth hormone gene of man into rabbit. In 1997, Dr. Lan Wilmot of Roslin Institute of Edinburgh, Scotland, successfully developed a clone of sheep, the Dolly, in 1979. Human growth hormones was synthesized in 1980. Paul Berg, Friedrich Sanger, and associates were awarded Nobel Prize of the discovery of recombinant DNA. The production technology based on genetic engineering is often called modern biotechnology. Interrelation of biotechnology with other branch of science. See the picture, the engineering, biology and chemistry, bioengineering, chemical engineering, biochemistry, that's called biotechnology. Biotechnology general information. Antigen, a compound which induces for the formation of antibody. Cellulose, wound tissue formed a wound. An undifferentiated mass of cells formed during the plant tissue culture, that's called callus. DNA ligase, enzyme which join the DNA fragments. DNA, the hereditary substance present in the nucleus which control the various metabolic activities along with the transmission of characters from one generation to another generation. A recombinant DNA, the altered DNA formed by manipulation in the original DNAs, constitutes of an organism using artificial method in laboratory is known as recombinant DNAs. Recombinant DNA technology, it includes those methods by which recombinant DNA is prepared. The method like insertion of DNA segments, reunion of segment by enzymes and fusion of these segments with Na of the other organism are included under recombinant DNA technology. Genetic engineering, beneficial changes in the heredity of an organism by genetic manipulation. Plasmid, an extra chromosomal circular DNA present in some bacteria. Restriction endonuclease, an enzyme that catalyzes the cleavage of DNA at highly specific site. Vector, an agent structure used for transfer DNA from one organism to another organism. Cloning, a process by which large number of identical copies or clones of a DNA or gene are developed. Explant, the plant from original source for the initiation of tissue culture on culture medium. Interferon, an antiviral proteinase agent synthesized by a tissue when attacked by a virus known as interferons. Bacteriophage, virus which infect bacteria. NIF gene, genes responsible for nitrogen fixation. Culture, microorganism grown or cultured on culture medium. Red biotechnology, medical application of biotechnology. Green biotechnology, application of biotechnology in the field of agriculture. Some important products prepared by the mediation of biotechnology and their utilization. Production and remarks. In the field of medicinal science, production of recombinant vaccine more effective and safe as compound to ordinary vaccine. Production of antibiotics, better protection from infectious disease, synthesis and production of valuable medicine, hormones and interferon, better management of non-infectious disease and metabolic disorders, gene therapy, diagnosis of hereditary disease in the field of agriculture, biological control of pest, biological control of weed, disease and harmful insects. Use of biofertilizer, protection from the harmful effect of this fertilizer and production of superior quality product, raising disease-free plant, high production with low cost, development of plant adapted to different environmental conditions, better ecological balance, use of burn and easy availability of plant products. Some important products prepared by the mediation of biotechnology and their utilization in the field: sewage treatment, proper management of sewage by the use of sustainable microorganisms, detoxification of industrial effluent, detoxicant by the use of microorganisms, production of nutritive edibles, use of many microorganisms and product edible substance, fuel production, production of biogas, ethanol, etc. Biopatent, the security of rights of personal property are guided by the laws prevailing in the country. In the present time, domination knowledge-based society is increasing. India is at position 1 in the queen of the knowledge-based society. The main basis of financial strength of such society is certain and preservation of intellectual assessed by them. Intellectual property can be in the form of a design, a concept, an idea, a research or any symbol. This property should be recognized as a personal or individual property, since its various forms are the result of mental capability of individual concern. For the security and recognition of intellectual assets, the protection by legal provision necessary. The legal protection provided by the government to these things is called a patent. The exclusive right granted by a government to an environment for the limited period of time to prevent other form commercial use of his invention, including a product or a design. Patents related to organisms and their components are called biopatents. In other words, biopatents are granted for biological entities and the products derived from them. Biopatents are granted for the following: strains of microorganisms, cell lines, genetically modified plants and animals, DNA sequence, the proteins encoded by DNA sequence process, some various biochemical process, production process, product and application of probiotics. In India, the variety of patent is for 20 years. The Patent Act made 1972. Bioethics. Ethics include a set of standards and norms by which a person or community regulates its behavior and decides as to which activity should be done or not. Therefore, bioethics can be considered as a group of norms and standards on the basis of which our behavior and vision towards the biological world can be regulated in biotechnology. Particularly, recombinant DNA technology is centralized over the exploitation of biological world by various methods. Plant tissue culture. The growth and maintenance of plant cell, tissue and organs on a suitable culture medium in vitro in a container, example test tube, is called plant tissue culture. A plant cell, tissue or organ exists from its original source and cultured in vitro is called explant. Tissue culture is of the following main types: plant tissue culture, culture of seedling, embryo culture, culture of isolated embryo, organ culture, culture of cell or tissues, tissue culture, culture of isolated protoplast. Types of plant tissue culture. The type of plant tissue culture are mainly based on the type of growth, example callus or suspension culture, or the type of explant from which the culture is initiated, example embryo, anther-sac, protoplast culture. Callus culture. Callus is an unorganized group of cultured cell. It is growing on agar culture media. Agar is a jelly-like substance obtained from seaweed, example algae, for a substance like axine, 2,4-dichlorophenoxyastic acid, 2,4-D, and cytokinin, benzyl aminopurine, BAP, are commonly present in culture medium. When the explant is kept in culture media, its cells being to divide and an unorganized mass of cells or callus is formed within 2 to 3 weeks. Suspension culture. In suspension culture, solitary cells or small group of cells are maintained suspended in the liquid culture medium. The explant is kept liquid medium. The suspension culture are necessarily agitated continuously by the electrically driven agitator at the rate of 100 to 200 revolutions per minute, so that the culture medium is continuously intermingled and the group of cells keep on back breaking into small groups. The growth rate of suspension culture is firmly high as compared to callus culture. See the developing callus in agar medium using for MS media. Taking explant getting for surface sterilization is transferred to the culture media. Using for agar, that's called as a solid media. The another one called as a liquid media. The liquid media containing for without agar, the suspension culture. The liquid nutrients is there, presence for the big, that's called a nurse technique. The liquid medium, the cells are suspended, getting for group of cells. Once again, formation of callus. The callus is transferred to the field. The callus and suspension culture are used for the production of biochemical, transgenic plants, protoplast and regeneration. Explant having pre-existing meristem, anther, young embryos, protoplast. Types of explant. Explant culture on agar culture medium, culture on liquid culture medium, callus culture, suspension culture, formation of callus, suspension of cell, regeneration, shoot, somatic embryo, emerging of rootlets, germination, plantlet, establishment of in the field. Use of callus and suspension culture, regeneration of plantlet, the cells culture in vitro culture finally give rise to complete plant, such as characteristic of plant cells is known as totipotency. During regeneration, the cultured cells from different structure like shoot and root, BAP, that's called cytokinin, stimulate shoot regeneration, whereas N, naphthalene acetic acid and axin is responsible for the production of root. In fact, formation of shoots and roots is controlled by balanced activity of axin and cytokinin. The embryo formed by somatic cells is called somatic embryo. Establishment of plantlets into field. The plantlets from culture container are shifted and planted into field. It is done by a process known as hardening. During hardening, the plantlets before establishing in the field are first put under reduced light and high humidity for some time to make them hard and acclimatize to the atmosphere. Protoplast culture and somatic hybridization. The hybrid formed by the fusion of somatic cells of different variety or species is called somatic hybrid. The process of their formation is called somatic hybridization. The various steps of this method are: first, of all the cell wall of the plant cell is dissolved by the help of enzyme like pectinase and cellulase. After dissolution of the cell wall, the remaining of the cell is known as protoplast. The fusion between the protoplast of selected parents is called protoplast fusion. It requires a treatment by a solution of polyethylene glycol or by a very brief high voltage electric currents. The product of fused protoplast is cultured in suitable medium. First of all, the cell wall is regeneration, followed by division of the cell, and finally generation of plantlets take place. The plantlets are subsequently established in the field after proper hardening. Importance of somatic hybridization. Somatic hybridization provides an opportunity for the production of hybrids. Somatic hybrids can be used for transfer of genes and cytoplasm. Development of new plant varieties. The process of development of new variety of plants is a long and highly complicated. For the development of high yielding varieties, following important steps are involved. Generation of genetic variation. Change in the genotype of plants is now as genetic variation. It can be achieved by the following methods. Collection of germplasm. The total number of genes present in a crop and related species is known as germplasm of the crop. Germplasm is the matter by which improving varieties can be developed. Therefore, for successful breeding, a collection of useful germplasm is necessary. The collection of germplasm is done from various parts of the country and even from other countries. See the picture. The nucleus is isolated. Cellulose and pectinase, the plasma membrane is removed, getting for isolated protoplast. Production of protoplast by the help of enzyme, mesozyme, lysozyme, that like that. Uh, Procurement of protoplast by the help of enzymes. Part A, protoplast, the nucleus, parent B, protoplast. Polyethylene glycol is mediator for fusogenic agent, formation fusion. The nuclear fission is occurred. Once again, formation of cell wall and the regeneration of new protoplast fusion, new plantlets. Polyethylene glycol introduced protoplast fusion and formation of somatic hybrid cells. Somatic hybrid cells come to somatic hybrid plantlets. Establishment is free. Use of somatic hybrids. Mutation. Sudden and inheritable changes in the characters of organism is called a mutation. It can take place by change in the structure of chromosome and change in the number of chromosome. The factor or agent which include mutation are called mutagens. Mutagens are two types. Chemical mutagens, that's called as ethyl methyl sulfonate and sodium. Azide or physical mutagen, example alpha, gamma and ultraviolet radiation. Hybridization. It is the most commonly used method for certain genetic variation. A cross between the population or parents of different genotype is called hybridization. Polyploidy. The plants are eukaryotic which are commonly diploid or polyploid. A condition when the somatic cells containing more than two sets of genomes is known as polyploidy. Methods. Mutation, gene biotechnology, polyploid, development of gene variation, the germplasm collection, protoplasm fusion. Selection, evaluation, multiplication of improved seeds, distribution of improved seeds, various steps of development of new plant variety. Selection. Selection of plants with superior and desired characters and development of the next generation by their seed is called selection. By selection, characters of crop can be improved. Evaluation. A newly developed population and hybrids with regard to their quality and disease and insect resistance is thoroughly evaluated in India. It is done by ICAR, Indian Council for Agricultural Research, New Delhi. Multiplication of improved seeds. Increase the number of seeds of new variety is called multiplication. Multiplication makes it possible to distribute the seed of the improved variety to the farmer. In plant breeding, the meaning of seed is that part of the plant by which new crop is grown. The grains of wheat and paddy, tubers of potato and stem cutting of sugarcane are called seeds only in the case when used to new crop. Distribution of improved seeds. Seeds of superior quality are called improved seeds. So they are distributed to farmer and agriculture research session. Production of food from sea plantlets. In oceans, algae can occur on rock, coral rock and free floating and commonly in Fordic zone. The edible sea algae are also called vegetable. Marine algae are rich in magnesium, iron, calcium, vitamin, folate, riboflavin, pantothenic acid, and anti-cancerous lignin. Kelp. A marine is a algae is the richest source of iodine containing highest amount of the element in nature. Presently, Janan is the largest consumer of sea algae as food. It is also the topmost producer and export of edible sea algae. They commonly used as follow: nori, porphyra, homo, laminaria, kelp, Dirnea pinnatifida, Irish moss, Chondrus crypsis. Sea algae are a good source of protein. For example, nori containing 20 to 33% of protein of its dry weight, 17.5 times more vitamin C than orange, and also rich in iodine. Medical biotechnology. Introduction. Food and health occupy a prime position. Human effort along with the scientific development. There is no doubt that because of this reason only, there has been maximum research in the field of medicine and health. Many aspects of preventing, diagnosis and cure related to various disorders and complex human health problems are being dealt conveniently by biotechnology is increasing the reference to the following medical fields. Inflammatory diseases, metabolic and homeostatic imbalance, hereditary disease, treatment of tumor and cancer. In medicine field, biotechnology has been used in synthesis of new effective antibiotics, synthesis of low-cost steroid medicine and hormones, development of better and safe vaccine, decreasing the use of chemicals in the treatment of disease, better use of radioactive substances. The latest service of biotechnology in medicine field is stem cell research. The stem cell research in the main base of tissue culture, organ culture and cloning. Stem cell. Zygote gives rise to complete in is individual, that is single cell, containing the complete information about the development of whole body. This cell has the capability of developing into any other kind of body cell by undergoing the division differentiation. The cells which are the base of development of a particular group or tissues in an adult organism or during its embryonic development are called stem cell. Blood cells are formed from the stem cells situated in bone marrow called the hematopoietic cells. The stem cell which have the capability of developing into any other kind of body cell are called the totipotent cell, example blastomas. While the cells which are capable of developing into a new few special or a of cell callus are called pluripotent or homeopathic cells. See, the zygote is undifferentiated. A relationship between the differentiation and potent of stem cell. Zygote undifferentiated, the totipotent cell. A blastoma, partially differentiated totipotent cell. Homeostatic stem cell, incomplete differentiation, pluripotent cells. Blood cells, RBC and WBC, completely differentiated non-potent cell. Cloning. Cloning is a Greek word which means branch. Clone is a copy of a duplicate obtained by asexual reproduction from a parent, mother or father, which cannot be considered as their offspring. This process formation of clone is called as cloning. Stem cell research in the main base of cloning technique. Cloning is the process of development of group of cells, tissues, organ or a complete individual by division and differentiation of single cell. All the cells obtained as a product of this process of a same genetic constituents. These cells are exact replica copy of each other and they are called as clone. Adult DNA cloning or complete organism cloning. It is the attempt of produce duplicates of an organism through this process. Embryo cloning. This process of development of monozoitic twins or triplets. Biomedical cloning. In this process, totipotent cells are separated from blastula or is the earlier stage and are used for culturing type of tissue or organ which can be transplanted again in the person. Cloning is divided into three groups. Cloning of complete organism, embryo cloning or monozoic twin, biomedical cloning. Cloning of complete organism, animal cloning, plant cloning, microorganism cloning. Animal cloning, sheep dolly was a clone. Cloning is a method of producing many identical organisms. Dolly was produced from a single of her mother. It had no father. Its hereditary characters were exactly like its mother because it was an offspring of a single parent. Dr. La Woolmer of Scotland produced first mammalian clone sheep named Dolly in 1997. Plants or animals normally reproduce by sexual method. By clone can be produced from a single animal or plant. An egg cell is taken from a female and its nucleus is removed, enucleated. A nucleus taken out from a somatic cell of some other mother. It develops into a new organism. Self-cloning is possible because of the presence of totipotency in plants and pluripotency of animals. Many laboratories of Department of Biotechnology of Government of India reworking in this field. Some other main organization are National Institute of Immunology, New Delhi, and Central for Cellular and Molecular Biology, Hyderabad, etc. See the picture. Clone mother cell completely differentiated into cell. The nucleus is 2N. Egg mother cells, undifferentiated egg cells and the nucleus. Isolated nucleus into nucleus 2N. Nucleus N denucleated. Denucleated egg cells is come fusion of nuclei electroporation method. The nucleus 2N cleavage. Implantation of blastula in surrounding mothers. Adult clone. For processing of cloning in brief. Microbial cloning. A genetically transformed microbial cell produces as many duplicate copies and may times it divides. In a few days, the produce thousands of clone cells, each containing same original genotype. In biotechnology, many improved genetical transformation. Microorganism can be obtained in a large number by clone. The microorganism transformed by genetic engineering can be used for various purposes. Example, E. coli is helpful is production of human insulin, human growth factor and interferons, while bacteria Rhizobium meliloti is helpful incorporating NIF gene for nitrogen fixing in crops. Plant cloning. Many drought-resistant, insect-resistant and herbicide-tolerant crops have been produced by a gene manipulation for agriculture. Food produced by a gene manipulation in which desired characters have been incorporated is called genetically modified food. Many genetically modified food materials, example lysine rich, pulses and vitamin A rich rice are becoming important constituents of human food. Animal cloning is more difficult as compared to plant cloning. Biomedical cloning. Tissue culture. Tissue culture is a technique in which tissue or cell is separated from the body of the organism. Rose Hassan in 1970 started work in the field of tissue culture and organ culture of organism. They use frog tissues as a source because they have high power of regeneration and grow faster as compared to mammalian tissue. Tissue culture has significant contribution in cancer research and production of antiviral vaccine. Production obtained from tissue culture of organism. Nature of the product, name of the products. Enzyme. Urocase, pepsin, renin, trypsin, hyaluronidase. Hormone. Folic stimulating hormones and luteinizing hormones. Vaccine. Rubella vaccine, mumps vaccine, measles vaccine. Interferon, human interleukin 2 and T cell growth factors. Organ culture. Part of an organ or its process can be grown in artificial culture medium, but it has not been possible to culture and complete organ as yet. Lot of care has to be taken during the culture so that the tissue does not get damaged during the process. Embryonic organisms can be cultured easily, but the process of culture of adult organs is very complex and difficult. Special type of serum, free media and special culture chambers are required for adult organ culture. The embryonic organism can be cultured in a two way, in liquid media, in solid media. Implementation of cultured organs. The most important aspect in the technique of implementation of cultured organs is there should be similarity in the genotype or the donor and the recipients. More the similarity in genotype, higher will be the success rate of organ transplantation. In transplantation, the normal response of the recipient is to reject the external factors because of the presence of many antigens in them. At the time of organ transplantation, the immune system of the recipient may consider it as foreign body and may reject it and destroy the transplant organ. Therefore, immunosuppressant medicine are used so that the immune system may not given and respond against the transplant organs. The success of the organ transplantation depends completely on the histocompatibility antigens present in the body cells. Test tube baby. The test tube baby is the main achievement of biotechnology human welfare. In this technique, the ovum is obtained from the body of the female and fertilized by the sperm of the male. Under control condition, this whole process is completed outside of the body of the female in the laboratory. If the egg cell or ovum is correctly fertilized and the process of cleavage starts in the fertilized egg, then it is transplanted into the uterus of the female where it undergoes normal development. Test tube baby is a baby obtained by normal development in uterus of the female, but after in vitro fertilization in the lab. The first test tube baby of the world, Louis Highborn, was born on the 25th July 1978 in Britain. The first test tube baby of India, Harsha, was born in 1986. Artificial insemination. Artificial insemination is a process of biotechnology collecting the semen of the male animal, storing in the extremely low temperature in suitable media, and transferring it into the vagina of the hostress, female of the fertilized, is called artificial insemination. The main aim of artificial insemination is quick improvement in the breeds of animals. Methods of artificial insemination. The main step of artificial insemination are follows. Collection of semen. The semen of the male of high quality breeds of animal is collected by different methods. Examination of semen. The semen of male of high quality breeds of animals is collected by different for the number of sperms, their viability in the vagina of female, and their capability of fusion with the ovum. Semen preservation. For preservation of semen, dilute so that its volume is increased and can be preserved for a long time. The substances which make the semen diluted are called diluents or dilute factors. Main diluting factors are egg yolk phosphate and egg or glycine. The preserved semen can be transported from one place to another. The diluted semen is preserved at minus 190 degrees centigrade at low temperature in liquid nitrogen. That's called cryopreservation. Insemination of female animal. The semen is introduced in the vagina of the female by rectovaginal techniques or any other methods. It is important that all the instrument and equipment used in artificial insemination should be properly sterilized or disinfected. Importance of artificial insemination. It is follow advantages. The improved variety or superior breeds of the animals can be produced by this method, but possibility of such production is very little by natural reproduction. The cattle farmers need to keep a few males so that they get maximum advantage is maximum expenditure. Artificial insemination increases the capability of female to become pregnant. It provides superior breeds of cow which give more milk and sheep producing high quality wool. High quality offspring can be obtained by fertilizing a large number of females even if the number of superior males is less. Difficulties or problem in artificial insemination. Trained cattle farmers are required for the process who are not available at many places. All equipments or instruments must be sterilized and should be of very high quality, which is very expensive. The expertise of identifying a female on heat comes by hard work or long experience. Use of radioactive elements in medical science. The elements which have same number of protons but have different number of neutrons are called isotopes. Such type of atom emits many type of radiation and are called as radioactive isotopes. These elements emit alpha, beta, gamma rays along with X-rays and neutrons. Main radioactive substance useful in medicine. See the iodine, imaging, the thyroid function, tactinium 99 thyroid test, galenium 67 diagnostic test for internal infection, thalenium 201 test of blood treated of parastrophy muscles, cesium 137 tumor treatment in physiotherapy, iodine 125 physiotherapy, cobalt 60 tumor treatment, gold 198 blood cancer treatment. Application of X-rays in medical science. X-ray are actually electromagnetic rays. They are invisible and do not create a sensation in the person. When they pass through the body, image of internal part of the body can be obtained by X-rays, especially bones, jaws, teeth, kidney, lungs, etc. At present, X-ray are used in the technique like X-ray image, CT scan and fluoroscopy. The image of transverse section of any part of the body can be obtained by using CT scan techniques. Cancer radiotherapy. Radioactive substances are also used in cancer treatment. The cells of the tumor are more sensitive to the radiation as compared to normal cell. That is why radioactive substances are highly effective in controlling the tumor cell. In cancer treatment, radioactive substances are mainly used as follows. Palliative therapy. The main aim of this treatment is to provide relief from the pain to the patient in the terminal stage of cancer. Curative therapy. The main aim of the therapy is tumor control and its destruction. In palliative therapy, radiation are given by the mild dose so that they do not cause any side effect along with allergic effect. But in curative therapy, radiation are given in very high dose. That is why they result in many side effects, especially hair loss. Brachytherapy. Brachytherapy is another type of radiotherapy in which radioactive substance is implanted in the tumor itself. External tumor of neck and head, tumor is prostate glands, etc. Use of laser rays. The laser rays are used in microsurgery. In medical field, the effect of laser on tissue depends upon their wavelength. It depends upon the type of laser rays. The wavelength of light produced by the carbon dioxide laser in 10.16 micron, which absorb the water from the target cell, and its energy is very quick, converted into heat. As a result, the effect of laser light is highly localized in neurosurgery. Laser rays are used to destroy these brain tumors, which cannot be easily removed by surgery. Visual organ ion laser have moderate effect of the tissue, so they are used in the microsurgery of eyes. The disease tissue of the body can be removed by laser rays. There is no loss of blood in the surgery, not treating the tissue, and no anesthetic drug is given to the patient. This is also called bloodless surgery. Whole laser technique has made it easy to get rid of any type of urinary disorder without surgical operation. Recombination DNA technology. Biotechnology is a science of application of manipulated DNA in the field of human welfare. This definition underlines the importance of genetics in biotechnology. The development and application of manipulated DNA is called genetic engineering. DNA is the genetic material in all organisms. In recombinant DNA technology, the DNA molecules is manipulated by changing nucleotide sequence, joining the different nucleotide sequence, transfer and establishment of changing sequence in the suitable organism. The important outcome products of the above applications are recombinant DNA. When two DNA from different sources are united or combined, the result DNA is called recombinant DNA. Transgenic organism. When the genes of different species are transferred into the organism, the newly formed hybrid is called a transgenic organism. It shows all the characters of transfer genes. Gene therapy. It is possible to changing the genotype or genetic constituents of a cell or organism by the application of biotechnology by doing so the genetic disease can be treated effectively. Recombinant DNA technology, a technique which proves the opportunity of integrating the gene or DNA of two different organisms into one, is known as recombinant DNA technology. The organism which contain genes of other organism in addition are called genetically modified organism or transgenic organism. See the picture, organism A, organism B. Give the excretion of DNA by restriction endonuclease enzyme, a sticky end fragment AB, a recombinant of the DNA ligase enzyme. See, the A and B is combined for hybrid. That's called as a rDNA technology. The technique of recombinant DNA technology can be employed for the transfer of genes responsible for the synthesis of insulin, growth hormones or interferons factor 8 into the other organism, particularly in bacteria. Application of recombinant DNA technology, production of insulin from bacteria. When the B cell of pancreas fail to synthesize adequate amount of insulin, the person is said to be suffered from diabetic mellitus. Human insulin is obtained from bacteria, Escherichia coli. The genes responsible for synthesis of insulin have been incorporated into E. coli. For this process is called humulin. Important products of recombinant DNA techniques and their application. A recombinant DNA product and application. Human growth hormones. Replacement of partially synthesized growth hormone in dwarf men. Human insulin, treatment of insulin dependent diabetes. Blood coagulant factors, replace of blood coagulation factors in hemophilic. Blood corpuscles producing factors, stimulation of formation of blood capsules during anemia. Interleukin, increase in the activities of immune system. Vaccine. For the control of herpes, influenza, wolf cough, hepatitis and meningitis. Advantages of transgenic plants. Object, disease resistant plants, weed resistant plants, insect resistant plants, virus resistant plants, to reduce the loss of crop production by biotic factors as to increase the production. The second one, cold tolerant plants, drought tolerant plants, salt tolerant plants. Proper use of land-plant growth possible to adverse conditions. Nitrogen fixation, more crop production, low requirement of monos on fertilizers. Increasing in nutritional value, increase the nutritional value and stored proteins in crops. Storage capacity, storage of fruits and vegetables for long period is possible. Losses during the storage and transportation are minimized. Attractiveness, fruits and vegetables are more attractive with regard to shape and colors. For the production of transgenic plants, the desired genes with the help of suitable vector or carrier are transferred and integrated into a new plantlet cell. TA plasmid are largely employed as a vector for the production of transgenic plants. BT plants. These transgenic plants has been produced by the introducing a gene of the bacterium Bacillus thuringiensis into it, which is responsible for synthesis of toxin that can kill the caterpillar of insect. Recently, PT genes has been successfully incorporated in maize, cotton and soya bean. Transgenic animal. Transgenic animal in which a gene of other species or source is incorporated. The foreign gene is incorporated into the embryo of the animal. The transgenic remain active in embryo or it can modify the expression of other gene. Transgenic animals are produced due to the following advantages. Development of Economically advantaged animals. In some milk-giving cattle, human gene control the synthesis of human protein have been incorporated. Development of disease model. The transgenic animal are highly suitable for a better understanding of several diseases of human being. It helps in the treatment of disease. Example, various carcinogenic genes which transfer to the transgenic rats wants mouse to understand the development of cancer in man. See some application and advantages. Medical application, transplantation of organism of animals, the development and growth of human organism in the body of animals is easy that facilitate organs transfer in man. Production of supplementary nutrient substance. Essential protein are essentially easily made available by animal product. The availability of insulin, growth factors and potential shall become possible. Uh, Human gene therapy. The use of transgenic animal is possible in gene therapy. Agriculture application. Improvement of variety. Development of superior variety of animal which are faster growing, more milk producing. Improving quality products. Improvement in the quality and quality or milk, wool and meat. Development of disease resistant variety. Development of various disease resistant variety. For example, development of influenza resistant variety of pig. The transfer of foreign gene into the embryo of transgenic animal is done by sophisticated technique like DNA micro injection, embryonic stem cell mediated gene transfer. Gene therapy. Many diseases in human are of hereditary nature. Example, hemophilia. Some diseases are caused by mutation of gene. Such diseases are not cured by general treatment method. Hereditary disease can be completely cured by changing the genetic composition. Gene therapy include replacement or alternation of defect gene, substitution of normal gene in place of defect gene and inhibition of the activity of defective gene, etc. In the certain times, treatment for some hereditary disease like hemophilia, thalassemia, cyst fibrosis, certain categories of cancer, high blood pressure and muscles dystrophy, etc. It is possible for gene therapy. Gene therapy of hemophilia. Hemophilia is a genetic disorder in which the blood coagulant factors like factor 8, 9 are lacking. The patient cannot sustain even a small injury. The flow of blood from the wound continue and may cause death of the injury. Gene therapy can introduce the gene responsible for the production of factors 8 and 9 in the cells of the patient and 9 in the cell of the patient. Thank you.